Okay, so we're going to talk about cell division and the cell cycle. There are three types of cell division depending on which type of cell you are. One is binary fission, and that occurs in prokaryotic cells. Another is mitosis, and that occurs in eukaryotic cells. And the other one is meiosis, and that also occurs in eukaryotic cells. So you need to recall that prokaryotic cells, like bacteria, do not have a nucleus, and eukaryotic cells do. So we have asexual and sexual reproduction. In asexual reproduction, that is reproduction of an organism in which one original organism makes an absolutely identical copy of itself. In sexual reproduction, that's where we have an organism that combines genetic material with two different or individuals of different sexes. Now, how does that relate to mitosis and meiosis? Well, for starters, let's look at binary fission. In prokaryotic cells, they don't go through mitosis and meiosis, they go through binary fission, which is an asexual way to reproduce. So it occurs in asexual organisms to reproduce identical offspring, and it is a 5-step process to do this. So binary fission, first the circular DNA is replicated, a new cell membrane is added. As new material is added, the cell membrane is going to push inward. A new cell wall is going to form around the membrane, and then the dividing organism is split into two identical cells. So recall that the first step in binary fission is DNA replication. Prokaryotic cells have circular DNA, which consists of a single loop chromosome. There are also separate chunks of DNA that are known as plasmids. So on your drawing of the DNA molecule, I want you to write bacterial DNA in the first box. Of course, my pen is not working. Okay, so bacterial DNA, and then the circular ones are plasmids. Okay, so let's talk about prokaryotic and eukaryotic cells for a little bit more. Because bacteria are single-celled organisms, reproduction and cell division for a prokaryotic cell are the exact same thing. That's kind of wild, but they are identical. Obviously, in eukaryotic cells, reproduction and cell division are two different processes. In cell division, you're making more cells within an individual. In reproduction, you are using, you're going through the process to create offspring. So there is a difference. Okay, so why do cells divide? And I need you to write this in your margin because for whatever reason, I deleted this information from your notes. You need to know that cell division occurs for repair, it occurs to replace, it occurs for reproduction, to reproduce, and it occurs for growth. So please jot that down in the margin somewhere and say this is why cells divide. Okay, so now I want you to find the spot that says cell cycle. And this is where you're going to fill in these notes. Cells are capable of growing, reproducing, and programmed cell death. Okay, therefore they have their own lifespans called a cell cycle. Cell cycle is a very delicate process. Scientists have made great strides in understanding the cell cycle because cell cycle malfunctions lead to cancer. So you can see the difference between a normal cell and a cancer cell. It is a mutation. Okay, so now I want you to move up your paper, and underneath the bacteria that you filled in those two boxes, you're going to see cell division is the process, and that's where we are at on your notes. So cell division is the process in which cellular and genetic material from one parent is divided between two new daughter cells. Okay, so here is your parent cell, and this is the daughter cells. And I would like for you to write that on your notes. Each daughter cell has its own nucleus. Daughter cells immediately enter interphase and begin their own cycles. So once this cell is duplicated, this original cell has nothing to do with it anymore. Okay, next page. Pre-cell division. Before the cell divides, the following must occur. The DNA must be replicated, which means copied, so that each cell gets a copy of DNA. After that happens, new organelles and proteins must be created so that each cell is equipped for survival. You have to have those things or it's not going to live. Okay, so now we're looking at chromosomes. Chromosomes are located in the nucleus of eukaryotic cells. And what is a chromosome? Well, let's remember chromosomes are DNA, which are many genes and proteins such as histones. A gene is a segment of DNA that codes for a particular protein, and they are located on chromosomes. Chromosomes are made up of DNA. Histones are proteins that help keep the DNA tightly coiled. Okay, so you see all of these little bundles. They are keeping that DNA, that double helix, all together. So typically when you hear the word chromosome, you automatically think of that. What you're looking at is more specifically referred to as sister chromatids. Okay. However, chromosomes can vary in form, and that's the picture we're about to look at. So I want you to write down this word, chromatid, label your first drawing. And then you see you have the centromere right here, which is just the center point where they touch and connect. And now you have you have sister chromatids, and you have chromatin, which is uncoiled. That's a collection. It's just like taking a hose and winding it up and having it all coiled together. Okay, those are the three forms that chromosomes come in. So I used the word centromere a minute ago, and now we're going to define it in the box. A centromere is a center of a chromosome. It serves as the point at which the sister chromatids will attach. Chromatin is loose, it's dispersed, it's immature chromosomes. The cell's DNA is in this form for most of its life. And can you remember where it's found? It's found in the nucleus, but you will also find it in the nucleolus. Okay, so let's discuss the cell cycle. In eukaryotes, cells have very specifically timed steps of the cell cycle. The following steps are included. We have interphase, and that includes G1, S, G2. Those are all a part of interphase. Okay, so that is right here. And we have mitosis. And mitosis has prophase, metaphase, anaphase, telophase. And then we go into cytokinesis. Those are all of the phases of the cell cycle. So interphase is the longest part of the cell cycle. It's broken down into 3 subphases, G1, S, and G2. So if this right here is the cell cycle, you're looking at interphase being majority of this cycle. Man, I cannot draw on this pad at all. This is awful. I draw better with my mouse. All right, you get the idea. Kind of, hopefully. Okay, so G1 stands for gap 1. And during the G1 phase, the cell's carrying out its routine functions, it's growing, it's making new proteins and organelles. The nucleus is well defined and surrounded by the nuclear envelope. The synthesis or S phase comes along and DNA is replicated. So it's copied in preparation for cell division. The G2 or the second growth phase is when the cell prepares for division by forming what's called a microtubules and other organelles or molecules in the cell that may need to divide. After the cell goes through interphase, it begins mitosis. And that's the process where you get two nuclei formed. Each is going to have an identical set of chromosomes, and there are 4 phases or steps of mitosis. And then finally, we have cytokinesis. So this right here is the mitosis, okay, this small portion, and then cytokinesis is when it just kicks out of the cycle. Because what you're doing is you're splitting in half and you're getting 2 new cells, each with its own nuclei and DNA. And this picture right here is of a plant cell. And you see a bunch of plant cells. So you can see the boxes right along here. There's one cell. Okay, there's another cell. And in the center, you're looking at the nucleus. And then you get to see the DNA that's separating as the new cells divide. So all of these cells are going through different stages of mitosis. You can see the cells about to shift and become 2 new cells right here. It's a pretty cool thing to see. Okay, so the cell cycle, typically cells are going to spend 90% of the time in interphase, growing and preparing for cell division. So what I would like for you to do is write down on your chart, you have this diagram. I want you to fill out this information. So we need to put cytokinesis on the line, because that's what this arrow is pointing to right here. You need to put the letter M, or you can spell out mitosis. If you color code this would be even better. G2, S, and G1. Okay, so all of those need to be pointed out on your notes. So which pieces or phases make up interphase? What do you think? Interphase is G1, it's S, and G2. All of those make up interphase. And we said that the cell spends 90% of its time in interphase. It very rarely, it only goes into mitosis when it needs to divide. All right, so let's talk about some really strange words. Somatic cells and gametes. So somatic cells are body cells. For every cell in your body, except the egg and the sperm, those are somatic or body cells. And all of those cells undergo mitosis. Every single one of them. Your heart, your liver, bone, skin. If it's not an egg cell or a sperm cell, it does not, it will go through mitosis because it is a body cell. The opposite of that is a gamete. Gamete are the sex cells, and gametes undergo meiosis. Okay, so there is a difference. This is your whole body. That's what we mean by body cells. And your sex cells, those are the eggs and the sperm. So a cell can be determined to be diploid or haploid, depending on the number of sets of chromosomes it has. So a cell of an organism consisting of 2 sets of chromosomes is going to be diploid. 1 set is from the mother and 1 set is from the father. These are somatic cells. Our body cells are diploid. In humans, the diploid number is 46. We have 46 chromosomes. 23 we got from mom, 23 we got from dad. And we symbolize this as 2N. Okay, 2N. And the prefix di means, well, it means 2. Okay, in haploid cells, Well, that's the opposite. It's a cell or an organism that has only one set of chromosomes. It has half that number, and those are the gametes or sex cells. Those are haploid. In humans, the haploid number is 23. There are 23, 23 in your eggs and 23 sperm, 23 cells with eggs and 23, and we symbolize that with an N. So if you think hap, that's like half. Okay, so in mitosis, what is it? Well, it's an important step in cell division in which a new nucleus is created. It occurs right before cell division. Mitosis only takes place in somatic cells, only in somatic cells. So it's only your body cell. It's extremely important. Meiosis, on the other hand, is going to cut it in half. It's only going to occur in the gametes, which are your sex cells. Okay, and you're going to get half the number. So mitosis 46, meiosis 23. Okay, so now that we have gone into the basic definitions of what mitosis and meiosis are, now we're going to actually go into each phase. And it's a 4-stage process in which 2 identical nuclei are created before cell division. Okay, the DNA of the original parent must be copied so that each new cell has the appropriate number of chromosomes. If it doesn't, then something is going to end up going wrong. So there are 4 phases of mitosis. You've written this down already. And we memorize mitosis by calling it PMAT. There's really nothing interesting other than some people say, I pee on a mat. They say, I pee on a mat because I stands for interphase. Prophase, metaphase, anaphase, telophase. Okay, each stage leads to the eventual separation of the parent cell into 2 daughter cells. In multicellular organisms, mitosis results in the growth and repair of the organism. But in unicellular organisms, such as some fungi, mitosis results in offspring because they go through asexual reproduction. So only in multicellular organisms does it use for growth and repair. So anytime you have a cut on your hand, a cut on your knee, any cut of your skin is undergoing mitosis to heal itself. Okay, so in mitosis, the very first step, we have prophase. Okay, what's happening here? Well, the cell prepares for nuclear division. It's going to break down this nuclear envelope. The chromosomes are going to condense, and now they're visible. You know, they used to be like a long string, and now they're all bunched into our sister chromatids. They look like an X. Okay, the mitotic spindle is forming, and centrioles are moving to opposite sides of the cell. All right, so on your note sheet, I want you to write the word nuclear envelope. That arrow is pointing at the dotted lines going around the nucleus. It's a nuclear envelope. It's just like the cell membrane. It goes around the nucleus. You also have an arrow pointing to the centrioles. And then you have these little mitotic spindle fibers. Combined on your paper, it kind of looks like a spider. Okay, so remember, the chromosome has to condense. It spends most of its time in the nucleus looking like this right here, but it has to condense to this guy. I don't know, I tried to put a hat on him and that just did not work. So sad. Okay, I'm not gonna quit my day job to be an artist. So we move on to the next phase. Once the cell has gone through that, it's condensed, we have our spindle fibers. What happens during metaphase? Well, the centrioles are established on opposite ends of the cell. The chromosomes line up in the cell's equator, which is the middle, and they attach to the spindle fibers. And right here you have your mitotic spindle. okay, and you have your centrioles. Those are the two parts right there you need to identify, and then it's pointing to the sister chromatids lined up at the equator. All right, it's very important that you document in your pictures what it is you're looking at. Okay, so metaphase is over. Well, we're not done yet. We have anaphase. And in anaphase, the spindle fibers shorten and they pull the chromatids to the opposite ends of the cell. So it's literally they're here and then boom, they are yanked back. Okay, that is the whole purpose of anaphase. The centromeres separate, so the chromosomes are no longer attached. So these are the spindle fibers. They shorten, okay, and your chromosomes are separated. They're no longer joined together. They're like, I miss you. Wait, come back. But they can't. After anaphase, we go through telophase. And in telophase, the nuclear envelope reforms. Okay. The chromosomes disperse into chromatin. The spindle fibers withdraw. So you need to identify this one as the nuclear envelope, and it is now pointing to the uncoiled chromatin. So as you can see, it's really getting to a point where everything is returning to the way it was. The cell membrane begins to push inward, creating what is called a cleavage furrow. And that right there is a picture of a cell that is about to cleave. It's about to separate. It looks like a peach right there. Okay, so cytokinesis is yet to happen. We've gone through the stages of mitosis, and now we need to go through cytokinesis to end it. Cytokinesis means cell, kinesis means movement. So cytokinesis is the splitting of one cell into two cells. This is when the daughter cells physically divide. Okay, so we have that cleavage about to happen. Cleavage furrow is coming right here, and then boom, new cell. Boom, new cell. Skin is healed. All right, so let's look at some of these examples here and try to name these phases. So number 1 on your paper, if you said telophase, you are correct. Number 2, okay, look at your chromosomes. You see they are in the center. We have metaphase. Number 3, they are being pulled apart. So we have anaphase. Number 4 is just prophase because it's just kind of a clump in the center. You can see the chromosomes the chromatin is all unwound. So there we go. The results of mitosis. Once it's complete, we get 2 daughter cells, and those numbers are diploid. We said that already. All right. Let's talk about plant versus animal because it is different. Okay. In animal cells, upon telophase, the spindle fibers tighten around the cell membrane. Okay, so looking at this, it's tightening. Just picture thread going over something and tightening it, and you basically just pull it tight until it separates into 2 separate ones. You get your new cells that way. In plant cells, a cell plate is formed. Okay, and that divides the nuclei of the two. And you have 2 cell walls that are created, and there's your 2 cells that way. Now, let's check for understanding and see if we can point out the parts of this cell. So, can you point out the centrioles? They're right there. Okay, so draw an arrow to the centrioles. What about the spindle fibers? If you pointed to that, you are correct. It could be any of them. Okay, all of this blue, these blue, those are your spindle fibers. What about the chromatids? Okay, so any of these guys. 1, 2, 3, 4 times 2. These guys over here. And what phase is that? If you said anaphase, you are correct. All right, so we are almost done. We've talked about mitosis. Now let's go into detail on meiosis. And you're going to see that it's pretty similar with the exception of a few things. So it's the cell division of gametes, and it's going to have half the number of chromosomes. It takes your number and it puts it in half. It's going to occur in all sexually reproducing organisms, and it happens twice. We have meiosis 1 and meiosis 2. It begins with the germ cell, which contains 46 chromosomes, and it ends with 4 gametes, which contain 23 chromosomes. Okay, so we start with 46, we end with 23. All right, so what about meiosis and chromosome number? Well, every cell has a nucleus, and every nucleus has chromosomes, and the number of chromosomes depends on the species that you're looking at. So, for example, humans, we have 46 chromosomes, but if you're a chicken, well, bakak, you have 78. A frog is going to have 26, and a pea is only going to have 14. But, strangely enough, chimpanzees and potatoes have 48 chromosomes. So, why should we bother with meiosis? Well, we need to bother with it because when gametes combine, the zygote, which is your fertilized egg, gets half of its chromosomes from mom and half of the chromosomes from dad. Okay, zygote is a word you're going to need to know. Zygotes are diploid. And remember, diploid is 2N. Okay, so zygotes are 2N because you have 2 haploids. So we have mom, well, let's see, we have dad and we have mom. They're both N, and that equals 2N for your zygote, which is your fertilized egg. Whoops, let me erase that. Okay, so the chromosome number of the egg and sperm is cut in half to ensure that the zygote will have the correct number of chromosomes, because there are issues that we're going to run into, genetic disorders and conditions, when the chromosome number is messed up due to meiosis occurring incorrectly. Okay, so this is your fertilized egg. All right, so meiosis is similar to mitosis, but it produces 4 unique daughter cells. Okay, 4 unique daughter cells, and each of them are haploid. So here is mitosis. You go through the stages, boom, boom, 2N, diploid. You go through the stage, separate, you go through the stage again, you separate. So that's why you get 4 haploid cells versus 2. Because we have, it's like mitosis 1, if there was such a thing, it's just mitosis. So this right here is meiosis 1, and this right here is meiosis 2. So genes are located on the chromosomes, and they control the individual's physical traits. They control your blood type, your eye color, you name it, they control it. Chromosomes come in matching sets, and we call them homologous. Okay, this right here is a karyotype. All right, and it's a picture of your chromosomes. The color-coded pairs you see are homologous pairs. So you arrange them by size, you compare the lengths to them, and if the bands look similar, if they look like you're going to match them up, then you put them side by side, and then you figure out the chromosome number, and then you figure out if your DNA has any genetic disorders or defects to it based on looking at your karyotype. Okay, so let's talk about homologous chromosomes, aka tetrads. Chromosomes are paired during meiosis, and the pairing is called a synapsis. They consist of 4 chromatids total. They're visible during meiosis. 1 pair is inherited from the individual's father. 1 pair is inherited from the mother. So you have a set of paternal, you have your maternal. They're identical in shape and size and the gene location. They have same genes but may have different alleles, and we'll talk about alleles in the future. During meiosis, they swap their genes or they cross over in order to diversify the gametes. Okay, so here is a picture of homologous chromosomes, and you've got all of these inherited traits. Okay, there's your alleles. And I know you know what I'm talking about. If you think big B, this is little h. Okay, and this one is Z or whatever. All right, so crossing over is a process in which homologous chromosomes exchange genes. So let's just look at this picture for a minute. You have your homologous chromosomes here. You are here. And then the chromosomal points at which crossing over occurs is called the chiasma. Okay, so this location is your chiasma, or chiasm. I don't remember how it's said. Important for genetic variation, if you didn't have crossing over take place, everybody would look the same. And it occurs during prophase 1. Okay, so meiosis, like we said, is divided into 2 stages. All right, I need you to write steps of meiosis 1 here, steps of meiosis 2 on this side, or you can just put meiosis 2, you don't have to put steps. Also, there's no DNA replication that occurs between meiosis 1 and meiosis 2. Instead, the number of chromosomes must be halved. So we're looking at prophase 1 of meiosis 1. We're just going to fill this chart out really quickly and then we'll be done. Prophase 1, we have crossing over that occurs among homologous chromosomes. It increases the number of possible gene combinations. Okay, so instead of us looking exactly like our parents or looking exactly identical to one of our parents, now we have a little bit of each. In metaphase 1, we have homologous pairs line up at the cell's equator. Okay, so it's just like metaphase originally. We have anaphase 1 is homologous chromosome pairs separate. And telophase 1 and cytokinesis 1, the nuclear membrane partially forms. The cell divides into 2 cells, and now it is a haploid 1N cell. Oh, but we're not done yet because we have to go through prophase 2 now. Okay, the nuclear envelope is going to reform. Sister chromatids again, they're going to line up in the center. In metaphase 2. In anaphase 2, the sister chromatids are separated. So they're moved in opposite poles. Goodbye, I love you. Don't forget to write. Okay, and the centromeres do not divide in mitosis until the second division of anaphase 2. I think that's supposed to say meiosis. But you don't have to write it down, so you're fine. Okay, so telophase 2 and cytokinesis 2, the 2 cells are now dividing, and this is how we get our 4 cells. So 4 daughter cells are formed, each having only 1 chromosome of each homologous pair. All right, so this is not on your paper, but let's practice. Let's see what we got. In meiosis, it involves, what's it involve? Well, it involves 2 divisions in which 1 diploid germ cell divides into 4 haploid gametes. Meiosis is the cell division of gametes in which the number of chromosomes is halved. So, are the results from meiosis identical? Why or why not? No. No, they're not identical. Okay, so super fast, let's go ahead and let's compare binary fission to mitosis and meiosis. Binary fission, remember this is prokaryotic. Okay, so who is that we're talking about? Prokaryotic bacteria. All right, this is how bacteria reproduces. It clones itself. Okay, so binary fission basically makes a clone. It's all identical. But for us, for mitosis, this is eukaryotic. Okay, this is people, animals, plants. Body cells, remember body cells go through mitosis. And sex cells go through meiosis.